Welcome back, class. So we just talked about our T cells. You can tell me how cytotoxic T cells can kill infected cells. And you can tell me how T helper cells can activate a B cell. But then what's going to happen to the B cell? Let's talk about what the B cells are doing. So some antigens are T dependent. They're going to present on MHC to a T helper cell and require a T helper cell to activate a B cell. Some antigens are T independent. They are able to stimulate a B cell to make antibodies without any T cell required in it. T independent antigens are going to be pretty weak, and it's really just big old pathogen interacts with antibodies, activates a B cell. But whether it is a T dependent or a T independent antigen, the B cell will then undergo clonal expansion. That means that B cell is going to say, woohoo, I am a good B cell. I found my antigen that I can attack, and it's time for me to multiply. And it'll multiply and multiply. It'll proliferate into a lot of B cells. And some of those will then stay around as memory cells to create antibodies for the rest of your life. And some of those B cells will very quickly start making a lot of antibodies, so you have lots of antibodies to attack the infection that's right there. When there is T-dependent antigens, let's talk about this a little bit more. So here is your antigen-presenting cell, a dendritic cell or something. It has found an antigen. It is now displaying the antigen on MHC class 2. So here is its MHC class 2 with the antigen on it. The T helper cell is able to come along and recognize that MHC class 2. When it recognizes that, this helper cell knows this is not a class 1. This isn't an infected cell. I don't kill it. This is an MHC class 2. It is teaching me that there is an infection here. And so then it is going to make cytokines to go off and activate a B cell. The B cell will then start making antibodies and come back and attack the antigen. So the B cell here is going to start interacting with the antigen fragment. The T helper cell will activate it. So it's sending the cytokines to activate the B cell. And now this B cell is maturing into a plasma cell that can secrete antibodies. So once this B cell is activated by a T helper cell, it'll differentiate in these two kinds of cells: plasma cells, which are these active antibody producing cells, and memory cells. The memory cells are inactive, but will just float around the body in case you get exposed to that antigen again. They'll remember the antigen. So if you have not been vaccinated, the first time you're exposed to it, you're going to have to wait until you make plasma cells. But if you have been vaccinated, you'll already have memory cells available. And so the first time your body is exposed to the actual pathogen, it's able to just immediately just jump right into making antibodies. So here's kind of the process of clonal selection. You're making all these different B cells, and some of them are just never going to be activated. They're never going to interact with an infectious particle, with an antigen. But here, this number 3 it has found a pathogen that it has found an epitope it can interact with. Some of them are going to be made into memory cells, and these memory cells will float through their immune system. And some of them are going to be activated into plasma cells. Those plasma cells are going to start making antibodies. Okay, so now you can tell me, plasma cells are producing antibodies. Are they also producing memory cells? Where are memory cells coming from? And what is the outcome of clonal selection? Then, when our antibodies and antigens are binding to each other, they form what's called an antigen-antibody complex. Sometimes those bonds are going to be really strong. The shape of the antigen and the shape of the antibody just fit together just perfectly. Sometimes they're going to be a little bit weaker. They kind of fit, but not the greatest. How strong that bond is referred to as affinity. And specificity refers to how specific the shape of the antibody is along with the antigen. Then when you have the antigen binding to the antibody, there are 5 different ways that organisms or toxins can be made harmless: agglutination, opsonization, complement fixation, cell-mediated cytotoxicity, and neutralization. Let's go through and talk about those 5 things. Agglutination means that things are clumping together. So because antibodies are bivalent, they can combine things together. So here you have multiple antibodies working together. So just like if you imagine you are cleaning up a room where a bunch of Legos have been spilled. One room, each Lego is broken up into individual blocks. And the other room, the Legos are in tall towers. It's going to be a lot easier to just pick up 5 or 6 towers of blocks than it is to clean up 2,000 individual little blocks. It works the same thing with our immune system. When they're clumped together, it's a lot easier for your phagocyte to just ingest and break down those bacteria when it's just dealing with big clumps than if it's trying to find a bunch of individual pieces. The next thing that antibodies do is opsonization. Opsonization means the same thing for antibodies as it did with complement. It means coating the antigen. So when that antigen is coated with antibodies, it's a lot easier for your phagocyte to interact with it. Your phagocytes can bind with the constant regions of the antibodies, and it's just able to quickly grab it, ingest it, and lyse down that cell. We already discussed how complement works in association with antibodies. So antibody, the Fc region, can bind to complement. That'll trigger complement to form this membrane attack complex, that donut of death, that creates the pore in the membrane and breaks down the cell. There's antibody-dependent cell-mediated cytotoxicity. So cytotoxicity means killing a cell, but in a way that this type of is going to depend on antibodies. So there are some pathogens, things like protozoans and helminths, that are just too big to be phagocytosed. Look at the size of this worm compared to the size of your white blood cells. They're not going to chew up this pathogen. Instead, the pathogen can be coated with antibodies, and then your immune cells will interact with the Fc regions of the antibodies, and they'll secrete chemicals, and those chemicals will lyse or break down the cell. So this is involving exocytosis, the ejection of these cells, molecules such as perforin, that will be breaking down the target cell external to the cell. So here's a perforin coming in, and it's just going to break down that parasite. And then there's neutralization. So neutralization is when either a virus or a bacteria or a toxin are just covered with antibodies. And when they're covered with antibodies, they're not able to bind to a cell. So the virus can't bind to a cell and get in. The toxin can't bind to a receptor and create any toxic effect. They've been effectively neutralized. Even though they're still there, they can't do anything. So which antibodies may activate the complement system, and which antibodies are usually associated with agglutination? Okay, so we covered a whole bunch of stuff about how B cells perform in our bodies to protect us from infections. We've just got a very little bit of stuff left to cover in this chapter. I'll see you back in our last chapter 17 video. Bye.